Hello, good morning, everyone. Welcome to the third episode of the Blue Cloud Ocean Literacy webinar. I'm Rita Menezes, Communication Essential Leader of the Blue Cloud, and I'm very happy, along with my fellow speakers, to have you with us today to talk about the role of artificial intelligence on ocean literacy. As you may be aware, artificial intelligence is everywhere and also in marine science. So the purpose of today's webinar is to showcase what you can do, what can be done with ocean literacy to improve the knowledge we have about marine landscape. So we have different speakers involved on different projects to give you an overview of what can be done. But first, we are going to open this webinar to give the floor to Sara Pitonet-Gayerin. She's one of the Blue Cloud 2026 coordinators. She'll provide an overview of the Blue Cloud's role within the marine landscape. After that, we'll give the floor to Diksha Penenok Martinez. They are involved on a project called Imagined, and they are going to give an overview of how AI is being used in this European project. After that, we'll give the floor to Rainer Kiko from Geomar, who is going to provide an overview on the different activities involved where AI is being used. If you may be wondering when you should pose your questions, so as you can see on the agenda, we will have 2 moments for Q&A where will be dedicated to reply to your questions. You can pose your questions by writing on the Q&A chat you have on Zoom. Our speakers will either reply you by writing or using his own voice during these Q&A moments that we have. If you wish to ask the question directly to our colleagues who are presenting, raise your hand, and depending on the time, we may give the floor to you and you can talk to us. Going back to the agenda, after the first session of Q&A, then we give the floor to Catherine, who is involved in the May Odyssey project. And after Catherine, we have Zakaria to tell us how AI is being used on Edito. And last but not least, we have Tiago Garcia from Plus Atlantic and Aerogoose Ocean Literacy Working Group. He's bringing to us today a project maybe less technical, but also important, which is about critical thinking in the world of AI. As you know, AI is almost everywhere. It's very easy to produce content, but the fact the content is produced doesn't mean is accurate. So Tiago is going to talk a little bit about this topic. And after this, we'll have one last moment for Q&A and the webinar will be closed. So I think we can start the webinar, and I'm happy to give the floor to Sara Pitonet to talk about Blue Cloud. Sara, over
Thank you. Thank you very much, Rita, and thank you to all our speakers today. So we're very glad to have you here for this webinar, which is part of a series that we're organizing within the framework of the Horizon Europe Blue Cloud 2026 project. So for those who don't know about the project, actually is becoming a long-lasting initiative so far. It started back with a previous initiative in 2019 with the idea of develop a virtual research environment federating key e-infrastructure, virtual infrastructure, and data infrastructures coming from the marine domain. So we started with a pilot that was consolidated with an actual infrastructure with a follow-up project in 2023 that started in 2023 that allowed this virtual research environment to become really operational and grow with a number of services and virtual laboratories that you'll get to learn. Now we are in a third phase. So while the actual Horizon Europe project is ending in June, the virtual research environment, so the entire platform, has become a European EOSC node. So in the framework of a program that is called European Open Science Cloud, that is building a federation at European level of research infrastructures, tools, and services available in a federated way for the entire research community in Europe. Blue Cloud has now been recognized as the node European Digital Twin Ocean, supporting research and in particularly research in collaboration with the European DTO program and its underlying infrastructure called EDITO. So across these years, we have grown. We've grown as services, as a technological platform, but also as a community. And we now respond to a multiple challenge and a multiple mandate. So on one side, we are part of this EOSC Federation, so like a system of system for European research. But then thanks to the research that is actually performed within the virtual research environment, we respond with services and methodological approaches to the European mission Restore Our Ocean and Waters. And the phase we are at now is a phase where we are integrating with the EDITO, as I said, the operational infrastructure of the European DTO program. This is the, let's say, the vision that we're trying to bring just to make all our stakeholders understand how complex is this framework, how complex is the platform, the services that we are building, but at the same time, how much and how far we are going to make this interoperable and federated and accessible to a broad number of users in different contexts. The EOSC node European DTO combines core and generic services, so IT, computing, resources, storage, combined with workflow management and monitoring tools, catalogs that are fully aligned with the open access principles built in the EOSC Federation, but at the same time, it's been integrated with the DITO platform. So the services coming out of the 5 virtual laboratories that Blue Cloud is hosting, with the free essential ocean variables workbenches that are being developed, will be offered and will be part of a broader offer under the EDITO umbrella, together with EDITO-specific capabilities that are being harmonized and federated. So, of course, all of these aim at responding to societal impact and in particular, of course, to marine need of a better marine science. We do so working with European services like EMODNet Copernicus, with research infrastructures like EuroArgo, ICOSIMS, but many more. And we really aim with the model that we're building, so with a structure, a strong infrastructure combined with dynamic virtual labs where communities can meet and collaboratively work to develop new services, new data flows, new applications and models, we can play as a blueprint for other thematic communities in the European Open Science Cloud framework. So as Rita mentioned already, and I started with, so this is part of a series of training webinars that we give, which are part of a broader training academy that Blue Cloud has developed. So just go on our website, you'll find training materials as guidelines or how-to materials on how to access the services, and you can watch recording of videos. And you can also access a series of courses that we run in collaboration with OTGA, the Ocean Teacher Global Academy of UNESCO. Thanks to the collaboration with the leaders of the virtual laboratories in Blue Cloud and OTG18. They're all available for free. I just conclude by inviting you all to join us for the final event, which actually we don't want to call it final because we already know that all this will continue through the framework of the EUSCANOD European Twin of the Ocean. But we're happy to welcome you in Brussels on the 28th of May at Comet Louise. And with that, I stop and I'm happy to give the floor to our speakers.
Thanks, Sarah. For this overview now is, let's say, Dick's and Enoch's time to come into play. Thank you for being here.
I hope you can see my presentation.
Yes, not yet in presentation mode.
Yeah, it comes. It's coming.
It's coming.
Yeah. Okay. You can see it.
Okay.
Full.
Yes.
Yeah. Okay. Now I want to. My name is Dick Schaap. I'm the scientific coordinator of the iMatching project. That is already finished, but I want to show you some of the results and how we approach this project. The project itself, it ran from 2022 to 2025. It's a European project. And we looked at how to develop and how to operate an AI framework as a platform that others could also use. And this was built not from scratch, but we followed up, let's say, from earlier work as part of the AI for EOSC and also the AI for EU projects. And to demonstrate and to make to see if the platform works, we worked with several use cases, all in the aquatic domain, and also from micro to macro, because we're dealing with image analysis, especially convolutional neural networks. But we looked at micro to macro. So you can say macro is the sentinel or the satellite data, and micro is really microscopic data and everything in between with webcams, etc. So it's quite a nice portfolio. The approach was as follows. In the middle, you see the platform. So like I said, we followed up from an earlier developments in several projects. And that platform was further developed. And at the same time, we also set up a so-called competence center. And competence center is a group of people that are very experienced in AI technologies, and they know very well the platform. And they interact with the use case owners to help them on their way to develop further, let's say, their use cases. And that's what you see on the top of the screen. You see the approach for the use cases. It's in fact, you start with data, you start with models, certain, let's say, AI models, and then you have several steps that you have to undertake. But this will be explained in more detail by my colleague Enoch later in his use case. And then also on the third level of the approach was that we have at several clouds, several, let's say, providers of resources. And you see some numbers there that we had 1,500 terabyte months, and almost half of them were consumed. But we also consumed about 365,000 GPU hours, and that's especially for the training. While at the same time, the CPU hours are lower, but they are more for the operations. Let's say once you have the model, you can run it, and that takes less, consumes less from the web providers were divided over Europe, so we had several. And this model we used, let's say, to develop several use cases. And we started in the project with predefined use cases, and we had 5 what we called mature. So people were already working on their models, and we tried to accelerate that and amplify that in this project. And also we had 3 prototypes. So people were having some ideas, they had some data, and they want to do something with that as part of this work. And as you can see, it's quite a diverse portfolio. So we have aquatic litter drones. So with drones, you are observing litter in the sea and on the beach. There is a marine ecosystem monitoring that will be later explained in more detail by Enoch. It works with cameras. Then we have FlowCam, which is a microscopic identification of phytoplankton. We have beach monitoring, which is done by webcams. We have ZooScan, again microscopic, with ecotaxa data. Then we have oil spill detection, that's more with satellite images. We have underwater noise, which is also, let's say, a new parameter or new indicator in the marine domain. And finally, we have diatoms identification. So quite a lot in the biological domain, but also some in let's say in the marine litter and other domains. We also had an open call running through the project, and by this we selected a couple of proposals that came forward by third parties, and in fact we helped them, let's say, to set up their use case again with the competence center. So they gave a lot of effort, let's say, to bring people on board, tell them how the system works, help them with going through the workflow, and then also the results. And I will not go in detail here because it will take too time, but you can see that's again quite a diverse operation and really gave us a lot to learn from this project. Everything is open and published. So there's the Imagine AI platform. There you can find both the modules as well as the tools. They are all available there. And if you click on, let's say, one of these boxes, then you can get a pop-up like you see on the right, let's say, for instance, for one of the use cases. But you can also go to do existing models that can be reused, let's say, for your purpose. Then we also published the best practices because we learned a lot from this. And it was done in a very nice deliverable, which is available on Zenodo. And as you can see on the right, it has already been downloaded and viewed many, many times by people. And the same best practice is also published as an article in Ecological Informatics. So it's the full document that has been published there. And that's another way, of course, to reach out to potential users and people that want to make use of the platform for their own applications. Because that's the two-way street. You can use the existing applications, or you can use it to develop and test your own applications using part of the pipeline that we have set up. Lessons learned. First of all, the AI-based object detection, so using images and AI to identify and classify what's on those images, that really can help and contribute to diverse aquatic problems and challenges. You need a lot of intensive support for new users because it's not so simple, let's say, to immediately grasp how it works. Annotating training data is very effort intensive. You need a lot of data to make good models, or let's say to have successful output from those models. And also another thing is that a good win is of course when these prototypes and these use cases that we have been developed, when they are really completely adopted as part of the operational pipeline that the institutes are using. And we had several use cases where this has happened. And that means, in fact, that the use case is sustained and being operationally used as part of the institute scientific workflow. Finally, we worked with several models. We tried out several models, and YOLO is an open source model which worked very well in most cases, and that was very popular, let's say, among our colleagues. Now, finally, what Imagine provides to you. So if you go to the portal, first of all, there is this common AI framework and computing platform, and you can use that for your options. There are several prototypes which are operational and which you can scan and analyze and learn from. There is this best practices document and training materials, and that helps you how to set up step by step. And finally, we have a collection of training data sets. In fact, for most of the use cases, all the training data sets have been published, and they're all published on Zenodo. And that's something that you can also use, of course, to try out yourself and see what you can do with those users. Now, finally, I want to give you a little bit more insight, and this is done by my colleague Enoch Martinez from UPC, and he will show you one of the use cases. And that's, like I say, it's very illustrative of what the project brought forward. Thank you. I'll give over now to Enoch.
Hey everyone. My name is Enoch Martinez. I'm from UPC, Technical University of Catalonia, and I will be showing this use case from the Imagine project. So I will go very briefly through what we want to do, which is ecosystem monitoring, how we apply the object detection, how we prepare the data, the training that we use, and the metrics and how we fine-tune our data, and finally, some scientific results. Okay, OPSI is a cabled underwater observatory, which is basically a junction box in the middle of the ocean with a power cable that goes to the shore, where we have power and communications, and we can deploy several sensors and instrumentation. It's in the Catalan coast, so north east of Spain, and it's very shallow. It's 20-meter depth, so it can be operated by scuba divers. Since 2011, we have deployed several cameras, and we have been acquiring underwater imagery for more than 15 years now. And why do we use this data? We use this data to study the fish abundance and the behavior of the fish, so basically biodiversity studies. But the thing is that analyzing underwater images by hand is very time consuming. What was the traditional approach to do this sort of biodiversity or ecological monitoring? So first we have the camera, we periodically take pictures, and then we had PhD students manually counting the fish that appear in every single of the pictures. And then they annotated all this into a spreadsheet. And finally, you could get a time series to work with your scientific data. Of course, this approach is far from optimal, as it requires students or professionals to spend thousands and thousands of hours counting fish in pictures. And this is what we want to automate. What we did in our use case in the Imagine project was to replace the human in the loop with an object detection model that could automatically extract the fish detections and classification from the images. And then we can store this into a database. And then we can do all sorts of fancy science with this data. So basically, the traditional approach is time consuming. It's not reproducible because different experts may not be may differ about the taxonomy. You need expertise in biology, and it's a very, very repetitive task. On the other hand, when we set up this automated pipeline using AI, first it requires a significant effort to set up. You need some strong IT skills, but in exchange, you achieve a fully automated workflow with no human intervention. So basically, we have now the scientists can do science instead of counting fish, which is a major breakthrough. So in a nutshell, what's the workflow or the whole process? So we divided when you want to deploy an object detection system, there are mainly 6 steps. The first one is you need to collect some of your data. Then you need to label your data because you need to transfer your knowledge, your own knowledge to a machine. And this is done by labeling your data, saying this is a sardine, this is an octopus. Then you need to select an object detection model. Of course, you can create your own model, but there is a huge community of open source models out there with a very, very high efficiency models. In our case, we chose YOLO. You only look once model. Then you need to format your data accordingly to your model. So basically some sort of transformation. And finally, you need to train the model with your data. This is where you need very intensive computational systems. So you need GPUs and this kind of systems to train your model so that your model learns from your data. And finally, you can use your model, what we call inference, and then you just send a new picture to your model and you get this sort of detections. That's exactly what we wanted to achieve. But now the question is, how good is this model? But let's take a look at the specific issues that we encounter in underwater images. In this case, for instance, you can see here a real picture with very dirty images. This is reality. This is what happens in underwater environments. You get a lot of biofouling, which is basically dirt in the glass or some sort of leftovers from fishes that can disrupt your image. And you have to deal with this somehow. So this is one of the main issues that you encounter. And then you also have other issues like when you don't have, even if you don't have this sort of biofouling into your optics, you can have also some very blurry images where you are not sure if if this is a fish or not. So are these fish in the background really background? Do you label them as fish? So there are some sort of tricky decisions that you need to make when deploying this kind of systems. Of course, there are all sorts of tools to prepare your datasets like Label Studio, RoboFlow, etc. So let's say the community to prepare data in machine learning is quite mature at this stage. And then once you label all your data, so of course if you don't highlight all your fishes, then you will cheat on your model and you won't detect all your data. Another issue that we encounter is what we call class balance. In real environments, specifically in biodiversity, you have very common species and very uncommon species. Of course, you can train your model with thousands of images, but you cannot train your model. In this case, we have one picture of an octopus. So your model will never know what is an octopus, which is just one picture. So you need to balance your data set somehow, and you need some sort of Solomonic decision on what to include and what to exclude from your model. In our case, our model will never detect an octopus because it's so rare that we don't have enough pictures. But for sure, we will detect, I don't know, Chromis chromis or Diplodocus sargus, which are the most common species. So next steps, we have our data set. We need to format our data, just change format, JSON to YAML, these kind of things. We train the model. In this case, our model can be trained using this simple command. And then we have the model and we can use the model. It works. We can see that it detects a lot of fishes, but another question is how good is our model? And then we need to take a look at the metrics. I'm not going to go into the details, but there are several metrics that will tell you how good is your model, like intersection over union, precision, recall. Are we detecting all the fishes? Are we missing some of the fishes? Are my detections correct? There's a whole field of metrics. And then you can have your nice curves to assess your performance. Okay, let's say that now we want to improve this, and we improve this, and then we apply it to get some results. So this is an example of some results using our model. We have been fine-tuning to achieve the best metrics that we could. This is one of the first deployments that we did of the system, and you can see here that we have a video, and all the fishes are being detected by the algorithm. This was a video mainly for let's say dissemination, but we have applied this to our time series, and what we get is actually time series. So we applied the system to time series of images, and we get a time series of a species. And this is exactly what we want. So we can now do scientific studies of the fish regarding of this time series, which is much simpler than looking at all the pictures manually. We have also did some experiments with artificial reefs in collaboration with other projects. You can see here, this is a man-made reef to restore the ecosystem, and we are assessing how the ecosystem is being restored based on the AI system that we deployed. We also had some studies with carcass of big mammals, in this case dolphins, to see what happens when there is a carcass of a big mammal in this case, and how this affects the ecosystem, and how the pattern of the seasonality and the species that are present or absent changes. And then you can do all sorts of biological studies much easier when you have these systems automatically processing your data. If anyone is interested in iterating our data, we have everything published in real time. It's machine actionable. We have an API. So if anyone is interested, they could potentially connect to our systems, download the data, and see all the detections, all the confidence, all the small nuances of our system in real time. So it's fully fair and interoperable in this aspect. And I want to close this presentation by just listing all our datasets and tools that we produced during the project, and they are available in Zenodo. So we have our training datasets, we have the pictures, we have the training ready to be, the dataset training ready for YOLO. We have our train model as well published on YOLO, and we have several scientific datasets with the data analyzed so anyone can use it. And of course, we are keep working on it beyond the project. So we will deploy new AI models soon, and we will aim to produce new datasets soon. Thank you for your attention.
You're muted, Rita.
Really, you're muted.
Yes. Now, can you hear me? Thank you.
Yes.
I was sharing some words of appreciation to the work you just presented. It's a real example where you can improve the speed how you do science. in different aspects, as Dick introduced. You had different use case, and the thing is, it was also a nice way to showcase, okay, we use AI, but how accurate is this AI when we were talking about the precision and so on, and that was quite impressive. And just looking at the comment from the chat, so it kind of is aligned with the last slide you showcased. People were wondering if there would be any source materials and references that they could check. So the last slide that you shared, Enoch, gives you some references and additional inputs if they want to consider, and use as benchmarks for their work. So thank you for this, Dick and Enoch, and to share your work on Imagine. And now it's about time to give the floor to Rainer and to share with us his experience. All good, Rainer?
Yes, I hope you can hear me well.
Yes.
Presentation is in presentation mode. Okay,
All
good.
good.
Yeah, thanks for the organizers for giving me the chance to speak. My name is Rainer Kiko. I work at the Geomar Helmholtz Center for Ocean Research, Kiel, Germany. And I would like to talk about AI for operational pelagic imaging and ecosystem assessments. I will present little result from PSSDB, a project that is already finished, and also mostly some motivation work for the Aqua project. Yeah, I would like to thank the Aqua team, the PSSDB team, Lars Stemmer, Marc Picheral, Jean-Olivier Risson. So what I'm going to talk about is plankton. So plankton are drifting organisms of all sizes, shapes, and colors. You can see some here. You see some very tiny phytoplankton organisms. Maybe here that might be a tracheodesmium organism, some copepods here, a little bit bigger, and then finally very big medusae. Everyone, I think, is a little bit familiar with this. So they are very beautiful, come in all sizes, shapes, and colors, and they have global importance. So what I show here is a map of the chlorophyll A distribution in the oceans, and you probably know that the phytoplankton that to some extent is depicted with this is responsible for, in over geological times, for the production of 50% of the oxygen we breathe. If we look at zooplankton and fish, they are the basis for 30% of the meat we eat. And finally, the atmospheric CO2 concentrations are 40% lower due to some ecosystem effects based on the interactions of phytoplankton, zooplankton, fish, and detritus than it would be without the so-called biological pump. So we have 200 ppm less in the atmosphere than would be without them. So these plankton organisms are important, and they have an uncertain future. We all know that the climate is warming, and this is actually due to the CO2 increase in the atmosphere. And the warming will also result in a decline of the phytoplankton concentration and the zooplankton concentration. At least models predict that. But somehow we know relatively little about zooplankton and phytoplankton because the methods we use are somehow cumbersome. So satellites actually give us nice global pictures, but they only can penetrate the first optical depth. So we know nothing really about stuff that happens at depth. And plankton nets can access deep regions, but the classical microscope approach to study them is actually very slow. So if we look at some data gathered over almost 60 years here about the zooplankton biomass distribution, you see, yeah, okay, it was covered. The world was, to some extent, most regions were covered somehow, but it took a long, long time. So we use a different approach from the classical one. So this is some stuff that I got engaged in 2012. Colleagues in Villefranche, they developed this underwater vision profiler. And I'm one of the users of these instruments. There are many others. This instrument can be deployed down to 6,000 meter depths. It's a camera system. In here you have the steel container that contains the camera looking down. And here at the bottom you have two lights that shine inward and generate an illuminated volume. This illuminated volume is imaged with 20 images per second, 20 hertz. And the objects that are on the images are then cut out and we use machine learning assisted sorting on the Ecotaxa platform to actually annotate these or classify them. So that's, so to say, an optical approach that I'm engaged in. And yeah, we have over years with many colleagues gathered the underwater vision profiler data set. So this is already a little bit older published work. In this work, we use 3,600 profiles, 8.5 million images in total, of which about 500,000 were plankton images. So they were all classified in a machine learning assisted way. Actually, it's still quite cumbersome. But we got biomass estimates from that. We couple that with environmental data, World Ocean Atlas, satellite data, and used boosted regression trees. So actually, again, a machine learning approach or AI approach, you can maybe call it, to develop such habitat models from which we can then predict the biomass. So that's actually one of the main outcomes of a lot of work. And here you can see maps of different organisms, copepoda, solidary colodaria, phaeodaria, krill decapods, and other crustaceans. So that's to some extent what we have done here and some published work. We have in the PSSDB project, which we've conducted in Villefranche together with colleagues from the US and funded by NOAA. We have then gathered more data and also used the more elaborate boosted regression tree approach to actually calculate the size distribution of zooplankton. So that here the map shows the size distribution of rhizarians, which is actually improved to just the biomass estimates, because when we know the size, we can, for example, calculate respiration rates in more detail. So yeah, this improved. We also have particle abundance or particle size spectra from all particles, and from that we can also calculate the carbon flux in the ocean, so the flux of sinking particles. And here I'm showing a reconstruction of, so to say, the global ocean distribution of flux. So this is a 3D picture. So it's just cutouts of the entire block, so to say, the 3D block of the ocean, visualizing flux in the different oceans here. So work done with Daniel Clemens and Daniele Bianchi, Thomas Weber in the US, and Lars Stemmann and myself. Yeah, so that's what's possible with the underwater vision profiler. And here my summary of this, so to say, motivation part of my talk. So pelagic ecosystems are crucial for humanity, and yeah, 30% of protein supply, 40% CO2 reduction, 50% oxygen. It's all based on the phytoplankton and zooplankton that we have in the ocean. Climate change, but also pollution, overfishing, etc., impact pelagic ecosystems massively. Classic microscopy and also, I think, sequencing-based observation techniques are too slow and maybe also too expensive. So we need to go for operational pelagic imaging assisted by AI to get such global estimates. If I want to summarize how we use AI in this workflow, then first of all, there's even at the sampling stage, we might even use AI because, for example, if we want to target eddies or fronts, we might use satellite data for targeting them. Then we need to do the image segmentation, the classification, and the exploitation. Yeah, and on all these steps, AI is being used. Main bottleneck, I think, is the classification of the image data. But the like exploitation, creating such global maps or also local maps, in my view, is impossible without machine learning and AI. So yeah, I've shown these showcases from the underwater vision profiler, but there are many other instruments in use by other colleagues and myself as well, that cover the size range of phytoplankton, microzooplankton, mesozooplankton. So we need to cover this size range. We actually need many different instruments. So that's clear, I hope. And yeah, there are also other projects like the IFCB, some imaging flow cytobots are used, for example, in the Gulf of Mexico for harmful algal bloom detection. We have seen some work from OPSI, and there's also CytoSub deployed there, which is used to image phytoplankton. We are now planning to deploy UVPs in the Med Sea, the North and Baltic Sea in the AquaSpec project, and in the Kimco project, we also want to deploy CytoSub and use also artificial intelligence tools to work with the data. Yeah, so but these are just some quick references to a few projects. There's much more going on in pelagic imaging. Yeah, and that brings me to the Aqua project, because somehow it's this map now shows where and how these devices are deployed, and you see that it's actually very diverse. And there are many units in use globally, but I would call this an incoherent approach because the observations are not aligned. So it's actually a fascinating challenge to contribute to operational pelagic imaging at global scales. And that's what we want to do in the Aquatic Life Foundation project. You have our website as well. So we have some challenges. As mentioned, the first one is that we have many different instruments, which also means we have many different data formats. And we have also each imaging instrument gives slightly different image types or styles. So you see here some with black background, some with white background, some image in the lab, some image in situ. You get different views, and still machine learning or AI solution should somehow tell that all of these are colanoid copepods, hopefully. So yeah, that's a challenge, different image styles, but also the different sizes and strong dataset imbalances that were also mentioned. So in the Aqua project, we have a huge team of programmers and scientists who work on digging into this data. We have a goal that we collect all images that were recovered here, that we bring them into a common data format and develop a foundation model. So that means that somehow everything is put together, that the huge computer crashes all the numbers and gives us some features and embeddings that are useful for fine-tuning, for example, the image classification. I don't want to go into more details. It's really just the motivation I want to bring here across why we are doing this. So yeah, we are funded since beginning of last year. We are in the middle of the project, and I really hope that we can release first products at the end of 2026. So here I would like to again share the vision of the Aquatic Life Foundation project. So we want to collect the image data from all different stakeholders, bring them together in a same data format. We will also publish the combined data set, but we will also develop tools to classify the image data, but also to map and monitor biodiversity, ecosystem health, and carbon flux globally, but also locally. Some data sets are really local, and yeah, that's to some extent our goal. Yeah, everything, our resources, data set, data format, the foundation, embeddings of the foundation model, but also, for example, an exploration tool that will enable the yeah, exploration of the embeddings and enable clustering of groups. Like the copepods will be used and made public so that everyone can benefit from these developments. So to summarize, I think it's a fascinating challenge to contribute to operational pelagic imaging at global scales. Yeah, if you would like to learn more about our project, please go to aqua.life. Or contact me personally via the shown email, or actually contact aqua@geomar.de. And yeah, you can still contribute data to this, but we'll also soon be able to use our resources. Thank you for your attention.
Thanks, Kiko. I have to confess, I enjoyed seeing so many different hardwares that you use in the project to collect different kinds of data. So, and it was quite nice, and crossing fingers for the remaining months of the projects. But before I look to the Q&A area, I just want, you were saying you have results in the following months because the project is rolling. So I'd like to know how the audience can be informed in a timely manner about your results. How do you share that information? Do you have a newsletter? So I think that's nice to know.
Yeah, we have some emailing lists via which we share our results. We have our website that you can visit and check for news. And yeah, we'll communicate it also with all the data providers, our status. So at the moment, we are still busy, setting up some tools, but everything will be shared.
Okay, thank you, Rainer. So I'm giving additional time for our audience. If you have a question to Rainer, to either raise your hand or write your question in Q&A. But in the meantime, while we are waiting for your additional questions, there is one. Actually, there are two. There is another one that was published while I was liaising with Rainer. So maybe Rainer, since you are here, we can start with you, and then we go to Enoch. So we have this question from Naweed about the global imaging maps. So the global imaging maps appear to suggest lower observational density across large parts of the Indian Ocean compared to other basins. So from your perspective, René, how could emerging regional capacity, human capital, regional partnerships, or even shared infrastructure could be mobilized to improve this pelagic observation and AI-enabled monitoring in this region?
Difficult and easy question. To some extent, of course, we have to follow this open source, open science approach, and thereby share data and make it accessible. And yeah, we also have to engage more in research in the area. Actually, the map I've showed, we can actually update it a little bit because at least from Germany, we've done a few research cruises last year in the Indian Ocean and just have to get the data online. That's ongoing work. So yeah, at the end, it all comes down to money. So one has to increase funding into this basic kind of research from all partners.
Okay, thank you, Rainer. And now, and a second question, but this time for Enoch, from our attendee. I think this is not the full name, MC Wilt, who really appreciates your work, Enoch, and he would like to know this. Have you had, Enoch, any success in isolating or managing individual fish so that one fish is not counted multiple times if it leaves the camera frame and returns back into the frame? Can this model be used for abundance as well as diversity? That's a nice question.
The short answer is no. We cannot identify fish, so we cannot assign an ID to fish. We just see detections of fish. What we can do, however, is to estimate abundance by counting the maximum amount of fish in a certain moment. For instance, if we have one day and I see at maximum we saw at the same time 50 individuals in a picture, that I know that at least there was 50 there. To go to biodiversity, to go to specific number, then you may need to have 360 cameras, which is much complex. Even if you have 360 cameras and you have low, let's say, if you have turbidity or low quality of the image, and the fish goes away and then would come back, it's difficult to really assign an ID because they all look basically the same. So we are working on that. We are working on achieving the maximum number, so we have some sort of estimated abundance. But we cannot have absolute numbers because you would need infinite field of view and you would need 360 cameras. But we can get a proxy of that. We are working on it.
Yes, in fact, I was thinking about the 360 cameras would maybe help, but still much higher investment but could be, you know, things. AI is being used quite recently, so we'll get improved and shaped in a different way. But thanks a lot for these possible compromise. And there is an additional question for you. About labeling, it seems. So we have Victor Garcia Pizarro, who is asking, labeling seems one of the most difficult tasks in the process you shared, Enoch. Do you feel using automatic labeling with models like Gemini, like Gemini? Oh, sorry, this skipped from. Oh, because there is another question. Okay, so do you feel using automatic labeling with models like Gemini can help to create initial bounding boxes for labelers?
I've never used Gemini for that, but could be. The thing is that you need to be sure that your bounding boxes are correct, because your model is going to learn from your data, and then you need the highest quality data. What we did was to have first an initial small data set, carefully handcrafted, and then we had some sort of semi-automatic labeling. So we trained a first iteration of the model, then we passed thousands of images to this initial model, and then manually we fine-tuned the predictions of the model. So we have some sort of semi-automatic labeling. I don't think that fully automatic labeling is makes sense, because basically if you have fully automatic labeling, then you don't need to train another model. But what we did is to have this sort of semi-automatic labeling where we train manually the first data set, you train, and then your first iteration of the data set predicts the bounding boxes for the next iteration of your data set. And then you can scale really quickly, because if you need to do everything by hand, it's a lot of work. But if you have this semi-automatic way, it can speed up the work.
Okay, thanks, Enoch. So I see we have two additional questions. For the benefit of time, I'm going to propose this. So Nawid and Violeta, I noticed you wrote this under the chat. If you write this on the Q&A area, so our speakers can reply writing. And then at the end of the webinar, after the other presentation, if we have time, we'll come back to these questions. But in this time, I'll propose you to write these on the Q&A so we can reply to you by writing, and we have to proceed with the webinar. And with that, I'm happy to give the floor to Catherine to talk about the Odisseia project.
Okay, so
Morning, Catherine.
morning. Oh, just sharing you, so you should see me in a few.
Not yet in presentation mode.
No, I think it's coming. Try again. Is it fine?
Yes, now it is.
Okay, so yeah, good morning, everyone. So today I'm going to present the Odisseia project, which is a project aiming at unveiling new hidden seafloor biodiversity by AI. So I'm working as an imagery and biology engineer in the deep sea lab in Ifremer. For people who don't know, fauna is a group of really small organisms with size range between 20 micrometers and 1 millimeters that live in the sediment in the benthic zone on the seafloor. They are known as the most diverse groups of organisms on Earth. They are quite abundant, and they were shown as good indicators of changes in the environment, and including for human impact assessment, because they respond rapidly to impact. They respond to low concentrations of contaminants, and we can find them in all types of sediments, from coastal to deep sea environments. However, they are also one of the most difficult group to study. Because technically the workflow to extract these organisms from the sediments is really it's really time consuming. So you see here there are different steps in this separation from the core sampling, then sieving, separation of meiofauna and organic matter from the sediment, and then hand picking and manual sorting and identification of organisms. So to improve knowledge in biodiversity and to accelerate this process, there is obviously a need to automate all this process and to classify massively all the organisms of the meiofauna. Also because other techniques such as fingerprinting methods are not really fully reliable, and also we are lacking references in depositories to link genes to species. So the initial idea was to develop automatic identification from imaging, and this using neural networks, also AI, to optimize the recognition. So the Meiodyssea project objective is to provide this fast, consistent, reliable, and standardized method for the identification of meiofauna. So providing high resolution morphological, but also functional and genetic information to accelerate taxonomy and biodiversity assessment. And this project, so it's ongoing, and we were able in this project to realize this initial idea of having instrument to automatically sort and isolate organisms. So that's the first instrument in the workflow. That's a cytometer called Copas Vision, and it is it's allowed to process really fastly and a lot of samples, and to produce large data sets of images of meiofauna. And after that, we are able to select specimens of interest and then to perform higher resolution imaging or barcoding and other techniques to produce additional and to gather additional information on the specimens, and even more to work on description of new species that we can detect. So in this context, we need, of course, to process and identify organisms on the large data sets of images produced by the cytometer. And for that, we based on work on previous big initiative around plankton that can have quite a similar range size as meiofauna. And we decided to use ecotaxa to classify automatically meiofauna organisms. So as it was already mentioned, ecotaxa is a collaborative annotation interface optimized to process large data set of images, and that provide tools for automatic classification that helps taxonomists for annotating organisms. And so it allows to import image dataset from different types of instruments. So in our case, we developed a program to allowing to transform raw data in thumbnails and object properties format compatible with ecotaxa. So we are able now to import images and object properties from the Copas in ecotaxa. And then in ecotaxa, we can either manually classify objects, but we can also launch a prediction. And firstly, we use the deep feature extractor of the ZooScan, as the images of ZooScan are quite similar to Copas ones. And that provide quite good results and help a lot in classifying our images. And after first prediction, we can validate subsets of images, then train again models and obtain scores. And in the second steps in the pipeline, we produce images from a microscope, Zeiss Axio Observer. These images are more heavy and complex than the one of the COPAS because it's a result of the projection of 26 stacks images. And so to be able to also to upload those images and analyze those images in Ecotaxa, we developed a software called EYES for embedded YOLO for every segmentation. And it allows to segmentate organism images and also to extract handcrafted features to be uploaded in Ecotaxa. And in the same way, we can then classify and predict automatically the types of organisms. And after that, to have a more specific deep feature extractor in Ecotaxa, so dedicated to meiofauna, we worked on images of nematodes produced by the EYES software and the handcrafted features. And that allows to develop so a deep feature extractor compatible with Ecotaxa. So using the ResNet model, resizing images and extracting the number of features that is expected in Ecotaxa. And we were able to integrate this deep feature extractor in a test instance of Ecotaxa in ephemer. And so that promise to make a more specific and more precise identification in the platform. And lastly, would like to present you an alternative way of working with this processing of prediction and validation of images in Ecotaxa. So we are planning to involve citizens in the step of validation. So the first after the first step of prediction. For that, we can export prediction and import them in a citizen science platform called SensePy, in which we have developed an interface dedicated to the validation. And so after validation by the citizen, we can upload again validated images in Ecotaxa and then ask time of experts to refine the classification. So this interface in SensePy is just the development is just finished, and it will be open maybe before the weekend or next in the coming days. To finish and to conclude, so we see that in so that was really a new domain in which we were developing methods using flow imaging and AI approaches to accelerate the classification of organisms. So we see that helps a lot, and by combining these approaches, these automatic approaches with AI and also manual and citizen validation, you can really optimize the speed of biodiversity data production. Of course, there is a need to adapt the development to the groups of organisms and also the different image modalities we have. And it is important to know that multidisciplinary work that is conducted together with IT and AI experts, also imaging engineers and researchers and taxonomists. So I thank you for your attention, and I will reply any question if you have.
Thanks, Catherine. Feel free to pose your questions to Catherine if you have any. Just a short comment. So far, the presentation, it was including one of your slides that is not of course, there is the work that you are doing within the project, but there was one slide where you mentioned citizen science. So part of also to use AI, citizens can be also involved through citizen science activities to help you classifying images and so on. So this collaboration can be even go beyond the people who are involved directly in the project, but also involve society on this, let's say, knowledge creation, which is quite relevant. Citizen science can also have an important role on this, and your project is one example of that. So thank you, Catherine, for your presentation. At the moment, I don't see any questions from you. They may pop in the next moment. So in the meantime, I'll give the floor to Zakaria from Mercator Ocean, who is here representing editor, who was already mentioned by Sarah Pitonet at the beginning of her presentation when she was going through the book audio review. Zakaria.
I'm just sharing my presentation. Can you see it right?
It's not yet in full presentation mode.
Well, let me try.
Maybe if you, it's the same. If you click on, you have properties on the right. If you click on the cross,
Oh yeah,
could help maybe. And it's not yet in full presentation mode. But if we cannot sort this out, this is a good compromise, I would say.
well, let me check. I don't know.
Let's proceed like this. I believe everyone can
Okay.
see.
All right. So hi everyone. My name is Zakaria. I'm a machine learning and software engineer at Mercator Ocean, and I'm quite happy to tell you today about Edito, and especially AI in Edito. So to be more specific, we'll go through forecast models, inference, orchestration, sorry, and as well as the benchmarking of data-driven ocean forecasting models. So for a bit of context, in 2024 was published the first major data-driven forecasting model, which was developed by a Chinese lab and which is named Chia. So they claim at the time that it produces good enough forecasts to challenge the best physical models, which is why it was very important for Mercator to evaluate this exact model, and which led to the decision in the end to explore on the possibility to train a data-driven model and see how well it could do, so a new one. So this was done by my colleague Anna Selauni, who is a PhD oceanograph with great expertise in machine learning. So to train this model named GLONET, he used the GLORIS reanalysis data over 5 variables, which are the salinity, the currents, sea surface height data, as well as the temperature, all interpolated on the 1/4 of a degree resolution, and as well as some atmospheric forcings added as an additional data and information for the model to correlate to the GLORIS data. So it's a multi-scale data-driven model. Why data-driven? Because it learns directly from the data and tries to capture the ocean dynamics directly from this data. So to sum it up, the architecture, it uses both spectral methods on the large scale to represent the large scale processes, and some convolutional methods to capture the more local features, so on a tighter scale. And the key is that it then combines both in a unified framework to forecast the ocean state. So in the end, the results were quite interesting, having better forecasts than the Chia model, especially for process-oriented diagnostics. So this led to the publishment of a paper. This was at the end of 2024. And then we started thinking about how we orchestrate daily forecasts of such a data-driven model right on Edito. So we have in Edito, which is the digital twin of the ocean platform, and especially in the data lab, the possibility to host and disseminate GLONET forecasts. Why? Because the inference runs of this model can fit on the Edito GPUs, which are resources that are provided to anyone, to the public. And using those resources, the first objective was to, in the data lab, create daily forecasts and produce them using the GLO12 nowcast, in Copernicus Marine, and to store the last 15 days of those forecasts at all times. Second objective would be to create the possibility to have on-demand forecasts for the public, so for anyone to create at any time. And last, some communication tools. So that's what we did, dedicating a project and some resources in the data lab. And right now, the result is that if you search for GLONET in the process catalog, you will find that. So 4 results. If we focus first on the GLONET forecast data for orchestration, so the second one, this is what I just told you about. It's an automatic process that every day fetches GLO12 nowcast data from Copernicus Marine, initializes the GLONET model with it to produce a forecast, and only the data for the last 15 days is stored, performing a sliding window. The other 3 processes are for on-demand forecasts, so for the public or anyone who wants to choose a date, for example, and create a forecast, or even create a forecast with more customization, for example, with the GLONET data init process, or even with the batch inference optimized process, which allows you to do inferences in batch, or using the GPUs that are provided through the Edito catalog. And then something quite important is that it's also available through GLONET's website page. So that's another channel to use if you want to create on-demand forecast. And the results of those inferences are in the ZAR format, making them light enough to be dropped in a viewer like MyOceanPro's viewer. So that's another way of visualizing those inferences. And then we might wonder how to evaluate the quality of such a model's forecasts. And this is another project named OceanBench. So that's a benchmark for data-driven global ocean forecasting systems. 3 main points that are quite important. First, that it's an open source project, so its code is public in a GitHub repository. So full transparency here. Also, it is open to contributors, so anyone can, for example, request for a new metric to be added, for example. And most importantly, it relies on the participation of any academic party that builds or built a data-driven ocean forecasting model, as we think that such a model can be easily added as a challenger, let's say, to our track, making the comparisons between models more complete, as we would have more models in our rankings. And also, quite importantly, this OceanBench was built so that its scores can be computed in an Edito Jupyter Python service. So what I mean by that is that you can open a Jupyter service in Python, install and import the OceanBench library, and compute the scores yourself at any time using the open source code. And those scores are displayed on the OceanBench website page, which is also hosted on Edito. To delve more into that OceanBench, so we have 3 tracks covered. So 2 tracks using physical models as a reference, those being GLORIS-12 pre-analysis and the GLO12 analysis, and 1 track using the observations data as a reference. So using the class 4 method for that, which is a method that allows us to compare the observations irregular grid to our regular grid data for the forecast. For the metrics, they all cover the 5 variables I just told you about, and go from a simple root mean square deviation between data points, so the most direct metric, to more process-oriented metrics, like for example, the deviation of Lagrangian trajectories or the RMSD of mixed layer depth. And on the right, I just show you some results, which are the results for the observations track. Personally, I think it's quite difficult to analyze such results in such little time, but I would say that the recent best data-driven models challenge the traditional physical models in a way that requires such detailed benchmarking tools to draw conclusions. So that's the legitimacy of Ocean Bench for me. And to finish, I would say that the recent AI developments at MOA, so Mercator Ocean International, for those, Editor Data Lab was a key tool in orchestrating AI forecasts and to compute benchmarking scores, allowing us to evaluate if the data-driven models forecast truly challenge the physical models forecast or not. And also, I think it's a very convenient tool if one wants to explore architectures applied to ocean data, especially for prototypes, which is a work that I did myself quite many times. And I thank you for your attention.
Thank you, Zakaria, for this overview of, from Mercator Ocean on the work implementing on 82. So for very full of time, I'll give the floor to Tiago now, and I'd like to remind the audience, if you have questions for Zakaria, feel free to write on the Q&A chat. So now let's hear from our guest, Tiago, who is going to talk about, it's important to have some critical thinking in this AI new world we are at the moment. So Tiago, you ready?
Yes.
Okay, lovely.
Hello, everyone. So we just saw how AI can help marine researchers to understand better the ocean and study numerous processes and issues that affect the ocean. So now I would like to propose shift the focus and think about how AI is shaping how the public at large is understanding the ocean and then affecting or being affected by the ocean. So, oops. Yeah. So to start things off, I would like to start with a little provocation. We produce all this knowledge, which is amazing. But how far we are in terms of being sure that anyone can understand the results that come from these projects. And when we talk about ocean literacy and the role of AI on ocean literacy in these days, are we sure that people are checking how scientifically correct is the content that AI generates? And who can actually indeed spot when everything goes wrong in terms of AI content? There's many examples out there. This is one that I saw maybe last year. Someone tried to reproduce what may have happened in Lisbon when the famous 1755 large earthquake and tsunami affected the city and most of the Atlantic basin, by the way. And the video that was produced by AI to reproduce what happened on that day looks very good. It's very entertaining. But when you know Lisbon, you start wondering if this is correct. And you will realize that there's many things that are not accurate, not only from the architectural point of view, but also even science aspects of the video. If you want to check it, I leave here the QR code for you to have a look. Where does the problem start? Honestly, I don't know. But since we're talking about researchers and research, I wonder what is the impact on society if a research center just says that one of their people published a paper? Does the public at large care about it? Are they going to access that paper? Are they going to read it? So all this knowledge that we are generating, are the citizens receiving it and using it to know more about the ocean? It's just something I would like you to reflect on. We already spoke about, it was mentioned in the beginning by Rita, that nowadays AI is being used a lot in everything, including in science communication and in ocean literacy. And obviously, as I mentioned, sometimes things go really, really bad. I don't know if all of you are aware of what a 500-year-old Portuguese caravel looked like, but I'm sure that this vessel here that it looks like it's made of steel and it's considerably larger than a caravel. I'm pretty sure this is not 500-year-old. However, this is online and people are believing it and following it and sharing this kind of contents. And if you want a more recent example, I think most of us were fascinated with what we saw happening around the moon recently. Lots of amazing pictures came out, legit, true pictures, but I have the feeling that this one is not one of them. Nevertheless, when I saw this, at least 400 people have put a heart, a like on this content. So we all know that the world is changing, the ocean is changing. Some of the projects presented before, they showcase how that change is happening. But my question is, what about the society? Is the society understanding this change exactly how it is happening? And when we think about education and its role on building citizens and the role of education on society, is the educational systems we have, are they ready to show this change in an effective and impactful way? Some people, when they talk about ocean problems, they tend to say that some of the issues happen in underdevelopment contexts, in underdeveloped countries, and come up with these narratives, which I don't agree at all. And let me tell you, what we are watching in this picture here, I've seen it happening outside of the European Commission buildings in Brussels during a coffee break of a very large ocean event. So if we cannot reach everyone, for example, within the European Commission, how are we going to reach the public at large? So with all of this in mind, I would like to introduce you to a new way of talking about science, promoting science education, and making sure that one day we will have a generation of people and families and friends of these people knowing about true science. So imagine something that all this misinformation would disappear. And for that, at Plus Atlantic, we teamed up with Escola Azul, which is the original Blue School program from Portugal. And we tried to fight all this misinformation, all this fake news, all this wrong content. We started doing this during COVID when we realized that there was a lot of science denialism going on about COVID and the vaccines and so on. And with this, I introduce you to Project 19. There is a music which I'm not sure you're listening to because I'm not sure I could share the sound, but nevertheless, the video is very short. I'll come back in a minute. So this is Project 19. As you can see, it combines marine science communication, science education, and the goal is to boost the ocean literacy levels within the younger generation, especially we're talking about teenagers in high schools, 15 to 18 years old. And what we do is we go to their schools, we give them research papers that have been published and cover very different marine matters. And then we ask them, what is relevant in this paper for you? And how would you tell a story about that? And we do try to go beyond the typical issues that people talk about when they think about the ocean, namely plastics and marine biodiversity. We know there's many more problems in the ocean. We wouldn't have a healthy ocean if we would suddenly remove plastics out of the ocean. There will be many other problems still there to be fixed. So with Project 19, we try to provide this broader perspective on all the issues that are affecting our ocean. And now what we learn with this is how to communicate to this generation. They are in a very critical phase of their lives. They're soon going to universities. Later, they will start their careers. They will start voting. They will probably become parents. So this is a very important moment of their lives. And how can we help prepare them to understand better science? And in this case, to understand when AI is not being true to science. So they told us that the way they want to be communicated, the way they want to receive this information is through all these engaging communication formats, artistic performances. They do think, and they did it actually, converted research papers into all these things, from theater plays, songs, from rock to hip hop to folk, videos, documentaries, dance shows, street arts, podcasts, and so on and on and on and on. So they actually converted research papers into this to tell their families, their friends, their teachers what is happening in the ocean. They told us that they had never heard about research papers before. They didn't even know those things existed. Now they know how to access them. They know how to interpret them. And they understood that if they want to make a statement or they want to defend a cause about something about the ocean, they know where to find real verified information. And this is very important in the times of AI. As for the teachers, they told us that even though we might not run Project 19 every year, they would like to apply the methodology in their school because, according to them, they never saw the students so excited and so engaged in learning about science. So this is amazing in terms of impact. And the project has been growing. We started in 21 in Portugal. Then we were invited to do it in South Africa. Then it happened in Brazil. And we are working already with Escola Azul, with the Blue School of Portugal, to have a new edition starting next September for the next school year. This project was endorsed as a UN Ocean Decade Action, which somehow also shows that this is something relevant, and more importantly, it's very replicable and usable by others. If you want to learn more, if you want to see the papers that were addressed in the first edition and what the Portuguese students did to convert them into their performances and storytelling and communication, you can visit the website. And of course, why not using this methodology for your own research, for your own papers, whether you are a PhD student or you are a director of a research center? Why not bringing it to the schools where you studied? Or if you are a professor, why not considering this to teach science, and in this case, marine science, in a different way? Or why not join forces with us at Plus Atlantic and let's put up a project to use Project 19 in a broader way. So that's it from my side. Many thanks, and I'm open for your questions. Thank you.
Thank you, Tiago, for this very nice presentation about, educating the future generations, because they are the future and they will shape the destiny, let's say, of our world when they will become adults. Very nice. And I'm looking at the time. We all reached our time we were supposed to have. I don't see any additional questions from our online attendees. So what I would like to say is everyone, so the presentations you saw today will be available on the event page. You'll be informed about that as well as the recording. And I would like to thank you for being with us today, for your interest in listening to the presentations we had. Also would like to thank your questions you made to our speakers, and naturally I would like to say a big word of thank you and appreciation to all the speakers who were with here today. It was nice to have this nice overview of the power, the good power, to take into account Tiago's words, Tiago's presentation, the good power of AI, how it can be used, how the AI itself can be improved, and we are very happy to have you all with us. So thank you, Sarah, Dick, Enoch, Rainer, Catherine, Tiago, and Zakaria for being with us today, and keep in touch. Have a good rest of the week.
Bye-bye.
Thank you. Bye.
Bye.